If you had all the money in the world, or at least some of all the money in the world, what other parts of the human body would you be interested in going to next?
Well, I mean, of course, there's obviously ethical issues. We just can't, take somebody's brain out to look inside. But
no, very tricky. Yeah.
But well, I think looking in the nervous system is important. I think looking in the gut is important. Those are probably the two things that I'd focus on more than anything. There are other tissues we'd like to look at, but they're not so common to get. For instance, we would like to look at lymph nodes, but most people don't have lymph node biopsies unless they have cancer or unless they're postmortem. So.
Yeah, I think wasn't lymph involved in the study of HIV in the beginning, or in the genesis of the?
Yeah, I mean, I think in HIV they had lymphadenopathy. They actually have expanded lymph nodes, so they probably.
Oh.
And some of them actually develop cancer too, so that would suggest that would be a reason why you'd want to do a lymph node biopsy.
Of course, people with ME tend to get what is it? Lymphoma that they get at an unusual rate, non-Hodgkin's lymphoma. Do you have any thought on that, how that might fit into the world as you've been able to illustrate it so far?
Well, actually, I kind of do. I'm not at liberty to talk about it because I don't want to disclose my research ideas, because if I do, then they'll be published before I can finish them, because I have a small group. So.
Oh.
But I can say that I think there is something to that, absolutely. I can give you some examples. So, like in our paper, we published that in a cohort of patients we worked with, and it was when I was working with Dan Peterson, Dan Peterson had identified that a lot of his patients had a clonal expansion of gamma delta T cells.
Who?
They had a clonal expansion of gamma delta T cells.
Okay.
So gamma delta T cells are basically a subset of T cells that are not CD4 T cells, or not CD8 T cells. They're kind of null cells. But he determined that in his patient population, they had this clonal expansion of gamma delta T cells, and those might be some patients who might be predisposed to lymphoma. And a lot of the patients who had lymphoma had the same clonality.
Oh.
Now, it's published in the research that expression of endogenous gamma retroviruses in animal models causes them to develop gamma delta T cell clonality. So it could be some relation. It could be, it may not be cause and effect. It might be just coincidence. It might be one thing is driving two things that happen differently. There's a lot of research that suggests that this type of dendritic cell is involved in some maintenance of lymphoma. It's an active area of research, in addition to the actual cell self becoming a lymphoma. But the cells are the primary producers of type 1 interferon. That's very important for the innate immune system. If you don't have a good type interferon response, you don't have good NK function. NKs are the killer cells of cancer cells. So a lot of these things are interconnected, so it's just up to us to figure out.
Yeah, I had really wondered whether the lymphoma had any connection stronger than the general tendency of people with impaired immune systems not to be able to fight as well. But you seem to think that there may be some more related, relatedness.
Yeah, I think that and that's actually Marion Fletcher and Nancy Kliman's work. The group has done a lot of work on NK cell function for good reason, because it's important. If you don't have good functioning NKs, those are the people who can develop lymphoma. That's what NKs do. They kill cancers, precancer cells. So it's an important area of research. I'm looking maybe a little bit farther upstream. Maybe what might be causing the NK dysfunction. It's possible this is involved. It may not necessarily be the only culprit. It may not even be involved, but it's conceivable. So,
I did want to ask about the brain. I know no one has ever done any research on my brain or any diagnosis at all. Yet I know that at least early on, people were doing a lot of SPECTs and PETs for diagnosis and so forth. But of course, as you say, you're very limited. You can't just go around plucking cells out of people's brains. Is there anything in your line of work that we can do about the brain, either through access to spinal liquid or through the imaging systems we have?
Well, from a researcher, I'm limited to the type of resources I can use, so
Yeah.
I don't have access to imaging systems, and honestly, if I had an imaging system, I'm not sure what I would do with it. Probably the same thing everybody else did, look for, bright objects and MRIs, things like that,
Yeah.
but then that's already been done, and what does it mean? People have looked in spinal fluid for pathogens.
Yeah.
I think Nielsen's group did looking for cytokines and found cytokine differences in spinal fluid. But yeah, I think that's probably the best we could hope for right now is looking in spinal fluid. One of the things that we're interested in expanding our horizon, because we are a neuroimmune institute, is to have diseases which have overlapping symptoms with ME, where it is more accessible to look at, for instance, like postmortem brain biopsies for people who have Alzheimer's, for instance. And we have those resources. We have an excellent collaboration. And that's when you mentioned the paper that had international people on there, it's because we have very strong international collaborations. In fact, one of our strongest, if not our strongest, collaboration right now is with a Russian group for the Institute of Medicine and Fundamental Biology at the Federal University of Kazan. So the big director there of the Genetics Institute was a former student at UNR and a close friend of me and my assistant Svetlana. And they're very much interested in things like MS and Alzheimer's, and so they have access to postmortem biopsies, and so we have resources doing work in collaboration with them that we may not have looking in ME patients. So for instance, if we identified something in, say, an Alzheimer's patient, then we might be able to say, right, how can we extend that into another disease that has neuroinflammation like ME or chronic fatigue syndrome?
Yeah, I feel that we haven't, in this case, I mean, as a nation, been doing enough to collect brains from people who have died. Of course, it's not officially recognizable as a cause of death, so that makes it difficult. But
exactly. Alzheimer's is a notable exception because that is absolutely a brain disease, and so having postmortem brain biopsies is not uncommon to get for Alzheimer's, but not very often do you get postmortem biopsies from people who die of ME. There's been a few people who have died. We do have some tissue that we're looking at, but they're very limited resources. And in science, if you do an investigation of 1 or 2 samples, it's not going to be accepted because it's not statistically meaningful. It could be just an isolated case, and someone might say, well, that person really didn't have ME. How do you know? But yeah, having access to those resources would be very valuable, but they're hard to come by, and obviously you can't get that biopsy if someone's still alive. So.
Yeah, I keep thinking that there must be something going on with the myalgia, are they? The specialized immune systems of the brain? Because headaches are so widespread.
Sure. Well, I mean, like I said, if you want to take a step back, you can say, okay, what is that really in the big picture? It's all inflammation.
Yeah.
And so if we can figure out what's driving the inflammation, that might be helpful. It's known that in at least in animal models, it's been shown that you can give a mouse injection of a stimulant like LPS, which is a bacterial product which causes an immune activation, and they can trigger a response across the blood-brain barrier without the stuff having to even go there. And so that type of inflammatory response could be triggered, for instance, in the gut, and could be transcended to the brain. So right now, in fact, I just wrote a proposal to look at MS, and that's the whole genesis of the proposal, looking at the brain-gut axis.
Oh.
In our paper, we mentioned that in a mouse model for MS, it's known that the gut can have direct effects on the brain.
Well, now what else is going on around your lab? What other kinds of things are you doing now and that you're writing proposals for?
Well, let's see. Currently, we are finishing up. We have 2 grants, a federal funded grant, a Department of Defense funded grant looking at chronic fatigue syndrome and Gulf War illness, respectively. Those grants we're doing, it's kind of a broad survey of the pathogenesis of the disease. So we're looking at genetics, we're looking at inflammatory markers. So it's very broad, but that work is ongoing. The Gulf War illness is basically a clone of the ME grant, so we're basically doing the same thing in Gulf War illnesses we're doing in ME because they're very closely related. The things that we're going forward on right now is the discoveries we've published recently about the gut and the dendritic cells. We're just going full steam on that and trying to understand that because that's the best clue we really have right now. although it's not practical to do gut biopsies as a diagnostic tool for ME, so it's not a useful biomarker, but at least it's a useful point to follow it, to understand what's going on. Even if it's not directly related to pathology, there's a difference between the patients and the people who are not patients. So it's meaningful, and if we can find out what the differences are, it might lead us to the next step, maybe to the next step, and then the next step, which could, maybe resolve the pathology of the disease. So it's an important clue, and like I said, when you're a detective, you follow the clues you have, and hopefully you find the killer at the end of the story, right?
Right. Well, next time I have an endoscopy, would you like a piece?
Absolutely, sure.
Really, maybe you should put out a general call for ME patients to...
Well, let's put it this way. We have human subjects protocols that say if a sample is used for clinical purposes and it's no longer useful, then we can accept it. So if anybody's had that, as long as your pathologist has used the specimen, then we could accept it.
Oh, right. Yeah. So I don't think anyone would go out and spend $2,000 or $3,000 just for fun, just to help out. But, yeah, if they are doing it. I mean, they did one on me about 2 years ago. I don't know if they would ever do it again. But, okay, so that's going on. And what are you writing proposals for? Is it strictly related to the dendritic issues or on other things?
That's our focus right now. So when it comes to ME or neuroimmune diseases, yes, we're looking at the dendritic cell angle and the gut issues. So there are some other things that, well, I'm writing a renewal for our existing grant, so that's ongoing. And then we just recently submitted an application for an MS grant.
And how many applications roughly, besides the renewals? How many do you think you have out? And
we've only got the one that's out in the system. We're writing the other one, and obviously you have to be somewhat focused, so you can't spend all your time writing grants. You have to do some lab work too. So
no, it's terribly hard to write grants.
It's very time consuming. So between writing papers, writing grants, and doing the lab work, it pretty much fills up an 18-hour day.
Yeah,
but, but so right now, MS, ME, finishing up the Goal for Illness project, and then possibly looking at maybe taking on one more project, maybe autism or Alzheimer's, something that's neuroimmune in nature. We've got some opportunities right now. It depends on preliminary data. So right now we're trying to generate some preliminary data that we can work off of for a grant.
And then that would expand you into other areas where funding might be more available.
Correct.
That would be very helpful.
Yes, and since these diseases are overlapping, things we learn from other diseases might directly benefit ME patients too. And that's kind of the thought behind all of this. Yeah,
that would be good. Well, I'm so glad that things are coming along, and I'm so glad that you came out with that paper this spring that was really launching us.
Oh, thank you. We have a couple more in the pipeline. We probably have 3 more papers we're writing right now. So
over the next year, they'll come out?
Probably within the next 6 months, if all goes well, yeah.
I'll just ask one other thing. Have you looked at any of the drugs that are being used or being proposed for MECFS?
Well, obviously, I was very interested in Ampligen.
Yeah.
The reason why I was interested in Ampligen is because Ampligen, if you understand the chemical structure of Ampligen, it's basically it's mismatched RNA. And
I didn't know that.
Right. It's a mismatched poly-IC, which is polyinosine, polycytosine, and so basically it's a big long RNA molecule, and it has some loops in it, which because it's mismatched, and those molecules are known to activate toll receptors. And the dendritic cell of my choice, which is PDCs, it has 2 toll receptors 7 and 9, and toll receptor 7, which is one of the toll receptors recognized as single-stranded RNA. So it's one of the, they're basically pathogen recognition receptors. And so for the most part, they recognize things that are not normally found in the body. And so, for instance, if you theoretically had a dysfunctional dendritic cell that had a toll receptor, it's possible that Ampligen might activate that. So that's something that we've had an interest in. But, unfortunately, since Ampligen wasn't approved, it's a little bit farther down on my to-do list because it's not going to generate funding dollars so we can keep doing our research.
Right.
But I do have some hypotheses about why that might be working. Clearly, Ampligen can activate NK cells, possibly through interferon. And obviously I'm interested in the rituximab study because the B-cell depleting drugs suggest autoimmunity, and that's exactly the direction we're going towards with our dendritic cell work.
Oh, that's interesting what you said about the amplogen, because I never really understood that connection. I knew it worked on the immune system, but I didn't understand what it had to do with.
Right, it most likely binds to toll receptors and activates toll receptors.
Good. Well, great. Well, I'm so glad to see you again, Vince.
Thank you.
And I'm glad that you're coming out with such great stuff.
Well,
and I look
to
forward to next year's crop.
Thank you very much.
Thank you.